Hi guys, today we're going to go through a little bit more on prokaryotes, comparing archaea to bacteria, and then spending some more time just talking about bacteria. So I know that you've seen this PowerPoint before, but I want to just go through it a little bit more deep with you so that you're ready for the exam. So this is a diagram that would be a really good essay question comparing archaea to bacteria, and this Venn diagram really shows the differences between them on either side. So archaea being what we consider extremists, that they like, phil, P-H-I-L, is liking or prefers. So this particular archaea prefers salt, halophile. Thermophile prefers heat and can live in very hot temperatures, and acidophiles can actually handle really low pHs and live in those kind of environments. Genetically, they're really most similar to us, which is kind of funny because most people think that bacteria would be similar to us, but archaea is actually most similar to eukaryotes. As far as classification is concerned, there's 3 to 5 kingdoms for archaea, whereas in bacteria there's 13 to 15. Similarities clearly are that they are prokaryotes, meaning that they're single-celled organism. But like we already did with our flashcards that we made, we know that there's other organisms that are also single-celled. There's some protists that are single-celled, and actually some fungus, specifically yeast, that can be single-celled. So although prokaryotes are considered single-celled, there are other organisms that can be single-celled. So they're the only organisms that are actually considered prokaryotic, where fungus and protists are actually eukaryotes, and that's from our flashcards. And we'll be getting, once we finish the viruses and prokaryote section, we'll be moving on to the protists and fungus. So that's when we'll be going from prokaryotic cells to eukaryotic cells. Prokaryotes also reproduce asexually, and that would also be on your flashcards that you guys made. So those are some differences between the two. One of the other things that I want you guys to be familiar with is the prokaryotic cell structure. So for each of these structures, to realize what they actually do, what's their function. Okay, the cytoplasm is the semi-solid liquid that all the cell structures are kind of floating in, if you will. The nucleoid, OID, is shape of, so it's in the shape of a nucleus, but there actually isn't a nucleus in a prokaryotic cell, and that's what makes a prokaryotic cell unique. So the nucleoid is where the genetic information is actually kept, and that genetic information powers everything for the cell. There's actually 3 different layers in a prokaryotic cell. The most inside layer is the plasma membrane, and here it's listed as a cytoplasmic membrane. After that, the cell wall, and in a bacterial cell, the cell wall has peptidoglycan, and that's an important thing when we get to staining, that particular layer can actually stain one color or another color depending on the type of bacteria you have. The outside layer is a capsule, and that actually provides a protective layer for the bacterial cells so that it can kind of adhere to surfaces and actually have a tendency to be more infective. The ribosomes, we know, are for the site of protein synthesis, and pili are actually used. This is an important thing to note, that they're not used for locomotion. Although they look like cilia, they're not. Pili are actually used to exchange genetic information between one prokaryotic cell and another, and so they actually create little bridges between each other, specifically in the process of conjugation, which we'll talk a little bit about a little bit later. Now, the flagella would be used for locomotion, so prokaryotic cell flagella is used to help that bacterial cell get around. So prokaryotes are classified in a lot of different ways. So the better we can classify a bacterial cell or a prokaryotic cell, the more we can actually communicate with each other clearly about that particular cell. So I would suggest making a mnemonic device so that you can remember all these different ways that the bacterial cells can be classified. There's really no word that this will form, and you can put it in any order you'd like. So if you wanted to just go alphabetically, you could start with the L, N, P, and then 2 S's, or if you want to just do the 2 S's first, whatever is going to work for you. But those are the ways that bacteria are classified. So shape, locomotion, staining, nutrient requirements, and pigments. So we're going to start off with talking about the shape. And here our prefixes are really helped us out here. So COCCI is berry, so it looks actually like a round berry here, and this is what this one is. And bacilli, is a little stick. So here we actually have something that looks like a little stick, and spirillum looks like a spiral. So how we actually classify bacteria is how they arrange themselves and by the shape. So if I were to say Diplococcus, that would mean I'd have two round prokaryotic cells. If I said I had a Staphylococcus, that would be like a grape-like cluster of round bacteria. If on the other hand I said I have a Streptococcus, that means that I have prokaryotic cells lined up in a chain, and they would be round. The other types we could have is Diplobacillus, Staphylobacillus, Streptobacillus, or I could have Diplospirillum, Staphylospirillum, or Streptobacillium. So these right here, these particular words are like our prefixes, and that tells us how those bacterial cells are arranged. And then the second part of the word is the shape. So Staphylococcus would mean cluster of round. If I said Streptobacillus, that would be a chain of rod-shaped or little sticks. So when people talk about bacterial cells, they'll say a certain type of shape, and then that tells us how that particular bacteria is arranging themselves under a microscope, so we could look at it under the microscope. We can actually classify bacteria too by how they move, and bacterial cells, the flagella actually does like a rotating action, so it kind of rotates around the central axis. So that's how the bacterial cells get around. The other way we talked about is the staining. And so we talked about earlier when I talked about the structure, the cell wall has this peptidoglycan layer, this orange layer here. And see how I've divided this slide up into the gram-positive, and then this is the gram-negative. So in a gram-positive bacteria, we only have two layers. We have the cell membrane, which is right here, and then we have the cell wall layer, and that's filled with this peptidoglycan. Now when physicians or bacteriologists stain bacteria, if it stains purple, they say it's a gram-positive bacterial cell. On the other hand, if they use a staining technique, and it's called a gram stain, that's where it gets its name. If it's a gram-negative bacteria, it has a cell membrane, just like the gram-positive does. It does have the cell wall, but notice how thin that cell wall layer is, and it also has peptidoglycan. But it has this extra layer here, and this extra outer layer is like that capsule layer, and it contains lipoprotein. So it has a little bit of a lipid component to it, and so it resists staining as well. So it has an extra layer. These bacterial cells tend to be more infective and require different antibiotics to treat. So these gram-negative bugs, we call those gram-negative bacteria, and they stain pink. So again, gram-negative bacteria tend to be a little bit more infective. People get a little bit more sick for those, and they're a little bit more difficult to treat medically. So we can stain bacteria again and review by shape, locomotion, and staining. We also can classify them by their nutrient requirements and pigments, and I don't have slides for those, but it's just something else to kind of realize. Bacterial cells reproduce by binary fission, and so this next slide talks a little bit about that and how quickly that can actually happen. So if conditions are good, bacteria can reproduce like crazy. So as long as they have what they need for their nutrients, the right temperature, whether it have oxygen or not, they can reproduce very, very quickly. So if you want to actually pause this section here and type in these two particular websites into another browser and watch these animations on how prokaryotes reproduce by binary fission, that would be great. And this is a method of reproduction that's considered asexual, so you're ending up with an exact clone. So why don't you take a couple seconds right now to watch those animations, and you might want to take some notes on that too, because another really good essay question could be to compare how a virus replicates, and we know that that replicates through the lytic and lysogenic cycle, lytic or lysogenic cycle, and then how bacteria reproduce. And so bacteria reproduce by binary fission. So knowing the differences of those would be important. And then when you're done, come on back. Okay, now bacteria do exchange genetic information a variety of different ways, and these are some ways that they exchange genetic information: by conjugation, transduction, and transformation. And under News and Edline, I've also posted some animations on these particular ones. Conjugation is the most popular one. Transduction and transformation do occur, but not as readily as conjugation does. And in the video that we watched on antibiotics, when they were talking how bacterial cells can exchange genetic information specifically to evolve to be multi-drug resistant, the process of that is through conjugation. So what I'd like you to do is make sure that you spend some time looking at conjugation and understand how they exchange genetic information through their pili, the sex pilus, to exchange genetic information so that they get different resistant strains so that they aren't affected by antibiotics. So this slide just kind of gives you a little bit of an overview on that, and if you take a moment to review those animations on Edline, that would be a great idea. Bacteria also can protect themselves for a variety of different ways. Slime layers are one, and that's with that capsule layer that we talked about earlier. And they also can produce endospores, which is under harsh conditions, they can produce these little spores. It's almost like a kind of concept of like going into hibernation to some extent. And this animation talks a little bit about that. So I would take a moment, pause the program, and cut and paste this into a new browser and watch that. And then when you do, come on back. Prokaryotes also form these symbiotic relationships. So if we use our prefixes here, IC is pertaining to, bio is life, and sim is together or with. So it's pertaining to life together. So different prokaryotes form these symbiotic relationships where everybody kind of wins. For us, they live in our intestines and provide us with B12 and some other vitamin K in our intestines. In animals, they also help to digest cellulose, and in plants they help with nitrogen fixation, so they have a lot of good things that they do for us. Well, not only good things, they recycle waste and oils in the environment, and here we would use the term bioremediation. So bioremediation is when we use them to kind of revitalize the environment. And Deer Island in East Boston is a classic example of that, where we're using prokaryotes to they actually help to make us fertilizer, and they get rid of sewage in the Boston Harbor. So that's one use of bioremediation, and we're using prokaryotes to our advantage. We also can use prokaryotes to make antibiotics and vitamins and certain medicines. Insulin can be made by bacteria, and so we can use prokaryotes to our advantage. It's also in our food sources, so if you like to eat a thing of yogurt, you can thank bacteria for that. And not only do they do good things for us, but they also can make us sick. So just like what we did with the viruses, I want you guys to know some differences of what is a bacterial disease and what is a viral disease. So you've got your notes from everyone's presentations on viruses, but I want you to go through that textbook and make sure that you know, I would say, at least 5 bacterial diseases. And some of those were in the video, like tuberculosis, they talked about that. And some other diseases that are caused by viruses, again, and some that are caused by bacterial cells. So that's kind of a nice little summary for you. I hope that helps you prepare for your exam, and at any point if you want to go back to any section, I would encourage it. All right, I'll see you guys soon.